So now we're going to look at the rest of the liver functions, but don't forget that many of these are ones that you've actually already seen. Now we're trying to put together some of the pieces and bring all these concepts full circle for you. So let's look at beta oxidation and ketogenesis. So beta oxidation, which is right here, is the process of utilizing fatty acids to make acetyl-CoA. That acetyl-CoA, which you can also see is formed in the process of glycolysis when we've broken down glucose or catabolized glucose, can then go into the citric acid cycle, also known as the Krebs cycle, so that we can extract the energy from those bonds and ultimately make large amounts of ATP. From a fatty acid, you can actually make about 108 ATPs. From a glucose molecule, you get about 36 to 38 ATPs. So there's much higher yield from fatty acids. However, if there is too much of those fatty acids, they can actually be converted to ketone bodies. That's a process called ketogenesis. That's this one right here that takes place in the liver as well. The formation of those ketone bodies creates biological acids. You don't have to know the specific names, but you do need to know the biological acids and acetone, which is the same chemical in nail polish remover, and you probably know what that does to your nails. Those ketone bodies can also lead to ketosis or ketoacidosis. So if you've worked with people who are diabetics or seen people that don't control their blood sugar well, you may have seen high levels of ketone in the urine or in the blood. You can also smell a sort of fruity scent on their breath as well. It smells kind of like juicy fruit gum, if you know what that smells like. That super sugary, nondescript kind of smell. It is common in starvation because in starvation, they burn through or catabolize all of their glucose, all of their glycogen, and then they go on to body fat. And then once much of the body fat is gone, then they will start to catabolize their own protein. So basically the body cannibalizes itself in starvation mode. Unwise dieting would be potentially things like the Atkins diet when it's not done properly, as in, eating a pound of bacon and a dozen eggs rather than eating high protein, low fat, low sugar extras in the diet, or again, in the case of uncontrolled diabetes mellitus. So if you do find high levels of ketones in urine, it isn't necessarily to say that the person has diabetes mellitus. There could certainly be an entirely different cause, such as liver failure, but it is one sign that practitioners might look at. In Chapter 17, we looked at hemostasis. And in the coagulation cascade, the third big step of hemostasis, we saw that at least 4 of those clotting factors, those are the ones with Roman numerals, need vitamin K to be synthesized. And most of those clotting factors are in fact made in the liver. And that is why liver failure can result in severe bleeding problems like uncontrolled bleeding or easy bruising that you might see in someone. So I'm going to save number 12 to go with 17. So let's go on to 13 here. The liver also stores some of our fat-soluble vitamins like A, D, and K. You can store those for months at a time. Vitamin B12 is also stored in the liver. It is B12 that is almost exclusively found in animal products, which is why you have to be careful with, your vegans, your vegetarians, that they're taking a B12 supplement. Some tofu companies have actually started to fortify their tofu with B12, which is a pretty smart thing to do, pretty smart marketing. And it's also why liver is sometimes recommended for people that have anemias, because it does store large amounts of iron and B12, both of which are required to make red blood cells. The liver stores glycogen, remember that's that storage form of glucose, and triglycerides, so it stores thousands of calories worth of those molecules. It synthesizes bile. That's its only metabolic function. So if you look at the digestive system and the role of the liver in that, the only one that's exclusively digestive is the synthesis of bile. And bile is important because it works like a detergent, which means it has some hydrophilic parts and some hydrophobic parts. So when it is bound to large globules of fat, it helps to break those up. And remember, this is a mechanical, not a chemical process, into smaller droplets. So the hydrophobic part attaches or is on the outside. The hydrophobic part is attached to the fat itself, to the fat molecule. The hydrophilic side is closest to the watery environment of blood plasma, and that helps us kind of dissolve. It's not really dissolving, but it helps to keep bile in solution, in blood solution, so that when you stand up, all of the fat doesn't go to your head, or when you lay down or stand on your head, it doesn't all sit or pool in one place. The liver is instrumental in helping us manufacture, synthesize, make hormones. So it does make some EPO, some erythropoietin. That's primarily the job of the kidneys, but the liver can help. And it synthesizes this prohormone, angiotensinogen. And angiotensinogen is that prohormone that's activated by renin to become angiotensin 1, which is activated by ACE to become angiotensin 2. And then the angiotensin 2 has multiple effects, even more that are in this picture, that help us elevate blood pressure if there's a hemorrhage, or if there's high potassium in the body, or if blood pressure goes down. So I know everyone is thrilled to talk about hormones again, but they don't go away. They're the second masters of control in our body, with the nervous system being the primary master controlling part of the human body. So the last set of liver functions that we'll talk about go back to chapter 17, where we talked about manufacturing and recycling of red blood cells. So remember this picture from chapter 17, and up here we had hemoglobin, and that hemoglobin is split into the globin chains, which are made up of amino acids. Those are the alpha and beta chains, those 4 chains that surround the iron-containing heme group. So the nitrogenous part of heme, which is the yellow part right here. The blue is the iron, but the heme part, or the part that's going to become bilirubin, is this yellow part on the outside here. So the bilirubin has to be processed. Some of it can be stored in the liver. Some of it is excess, so it ends up being excreted through fecal material. So it goes from the liver into the blood and then out through the rectum. And that is stercobilinin, which is sort of like a yellowy brownish color. It can also be excreted through urine in a molecule called urobilinogen, which accounts for some of the yellowy color of urine. Or some bilirubin becomes part of bile. So the iron part of the heme group can be processed and transported, or it can be stored in the liver. So iron can be stored in the liver, bound to hemosiderin or ferritin, which helps keep the iron cation from being reactive with other species or other ions, or you can bind the iron to a molecule called transferrin, trans as in transport, ferrin, Fe for iron, and then that can go into the bloodstream back to brain maybe, or it can go to the red bone marrow to make red blood cells.